Here we go. So I want to start going from our great peak where we are, where we've learned about DNA, RNA, proteins, transcription, and translation, to start going back down from the peak of insight down to the flatlands of understanding. And I want to go down by way of looking in more detail at cells. Now, we talked at the very start about how all life as we know it is cells or made of multiple cells. And so now we can actually look a little bit more at what you can see inside cells and what cells actually do. Any cell, prokaryote or eukaryote, contains a flowing liquid substance called cytoplasm. What you're looking at here is a large eukaryote. That's an amoeba. And it's a eukaryote. The nucleus is right there. There's various little blibs and blobs inside that we don't need to get into. And they're all floating inside a complex solution of water, salts, proteins, lots of other stuff. The liquid goo inside a cell, that is cytoplasm. Cyto, by the way, is Greek for a cell. I think it actually means a box. And what keeps the cytoplasm in and the outside out is called the plasma membrane. And the plasma membrane is not really a solid. It's not like rubber or anything like that. It's a two-dimensional film of lipids. You might remember from way back that we looked at the four basic classes of macromolecule: carbohydrates, lipids, proteins, and nucleic acids. Well, we've talked about proteins and nucleic acids until you probably never want to hear about them again. Lipids is the class that includes fats and oils, also waxes, incidentally. And a particular type of lipids, they're called phospholipids, if you must know. If you add them to water, they'll line up in a particular way to make bubble-like structures that are bounded by a double layer of lipid molecules called the phospholipid bilayer. In the interest of time, I'm not going to say much more about exactly how they do this, except to say that they do this spontaneously. You can make lipid bilayers by shaking up your salad dressing. And in a living cell, proteins and other molecules like glycoproteins, that's a protein with some carbohydrates stuck on it, can be embedded in the membrane, and some of them will stick out of one side and or they might stick out of the other. Some of these are transport proteins that allow things to move into the cell or move out. Some of these are sensory proteins. We've seen some of these before. They will bond with something else and bring it into the cell or cause a change inside the cell so the cell can respond to the outside. There's lots of different things that these can do. And again, the membrane is a very thin film of liquid. It's not exactly a soap bubble, but that's closer to it than anything else. It's not like a rubber balloon, which is stretchy but still ultimately a solid thing. And because cell membranes are films of liquid, membranes, not just the outer membrane outside the cell, but a lot of things that we'll see inside the cell, because they're all these films of lipids, they can easily fuse together or they can pinch apart. If you stare at soap suds for long enough, you'll see some bubbles that come together, and then in between them the partition between them bursts and they fuse to become one bubble. Or with a bit of stirring, you could actually make large bubbles split into many little bubbles. That's the best analog for how cells do it. They're able to divide and in some cases fuse, and structures inside them can divide and fuse and bud off and rejoin and all that because they are they're bubble-like liquid films. So there. Okay, we talked a long time ago about the difference between prokaryotes and eukaryotes. Most prokaryotes outside that cell membrane, they've got a cell wall. You could make a decent model of a prokaryote by taking a water balloon and stuffing it in a shoebox. The water would be the cytoplasm, the rubber skin would be the cell membrane, and the shoebox on the outside would be the cell wall. And in a typical prokaryote, there's kind of a central area where the DNA is located. All of this, kind of whitish looking, vaguely stringy, spaghetti-like stuff, that's bacterial DNA. And that area is called a nucleoid, but not a nucleus because it's not bounded by a membrane sac. It's just floating there in the cytoplasm. And so are the little structures called ribosomes, where a prokaryote translates messenger RNA to make proteins. So remember, in a prokaryote, nothing's really partitioned. Everything's kind of floating around in one big room. Sorry, just checking chat, seeing if we've got questions or anything, and admitting a couple of people. All right. Okay. I haven't said much about prokaryote DNA, but I should mention that it's organized not into chromosomes the way that eukaryote DNA is. Eukaryote chromosomes are long strips of DNA and they have two ends. Like any piece of string, they have, one end and another end. One of the differences between prokaryotes and eukaryotes that we didn't get into at the very beginning, but that I can reveal now, is that bacterial DNA comes in loops, not in strips. Typically, you'll have one large loop of DNA plus a number of smaller loops called plasmids. These are a couple of bacterial plasmids shown with the scanning electron microscope. They're just loops of DNA with maybe just a couple of genes on them each. And bacteria can swap plasmids like kids trading Pokemon cards. Bacteria can also just pick up plasmids from the environment. And this, in fact, is what was happening when Frederick Griffith mixed his dead S-type bacteria with his living R-type bacteria. The R's were picking up plasmids from the dead bacteria. That's what the transforming principle was. When bacteria have sex, they do have sex, although it doesn't quite look like what you're familiar with. What they do is one passes a plasmid to the other. So yeah, when bacteria have sex, they're basically genetically engineering each other. Hopefully you still remember that eukaryotes have a defined nucleus, which is stained here in this picture in blue. And the nucleus, I seem to have spelled it mucleus. That's only in cows. Just kidding. That's a nucleus bound by a double-layered nuclear membrane. So there's an actual lipid membrane sac between the DNA in the nucleus and the cytoplasm filling up the rest of the cell. This happens to be a plant cell nucleus. All of this round-ish thing that's filled with grainy material, that's the nucleus itself. And then inside the nucleus, there's one, or occasionally there might be more, areas that you can see even under a light microscope as looking dark. This is it right here. This is called the nucleolus, and this happens to be where ribosomes themselves get made. That's where the cell can manufacture ribosomes, which of course pass out of the nucleus into the cytoplasm where translation takes place. Just in general, I'll point out that this is a eukaryote, so you can see it's compartmentalized. You've got a nucleus here. You've got a bunch of little compartments over here. Those are called mitochondria, and we'll talk about them in a bit. This thing right here is yet another compartment called a chloroplast, and we'll talk about that in a bit. Although I think you should have seen them in the microscope lab that you did. There's another one of these compartments. You know, even if we haven't covered what all of these compartments are, you should be able to see that the eukaryote cell is much more compartmentalized. Okay, what you're looking at is the outer surface of a cell nucleus. This has been taken with a technique called freeze fracture microscopy, and the nucleus is right there, and it looks like, it frankly looks like my face when I was about 13. You can see it's got a whole bunch of little dots in it right there. Those are called nuclear pores, and they're held open by some rather complex proteins called porins that act as gates. The nuclear pores can open and close, so that's how ribosomes can get out of the nucleus and go to where they're needed. Messenger RNA goes out of the nuclear pores. Certain proteins might pass into the nucleus via the nuclear pores. So that's where traffic in and out of the nucleus is controlled. So what's the rest of the cell full of? Well, it's full of cytoplasm, but we now know there's a lot more structure than that, most of which you can't see with the light microscope, or at least not. You can sometimes see a little bit of it with staining, but a lot of this we didn't know about at all until the rise of more powerful forms of microscopy. So we now know that a good chunk of the inside of a cell is filled up with a network of sheets and folds and tubes and pipes and blibs and blobs of membrane, folded up and rolled up inside the cell. We call the whole thing the endomembrane system, and by far the largest part of the endomembrane system is called the endoplasmic reticulum. And if you don't feel like saying endoplasmic reticulum, we can call it the ER. This is basically the cell's main transport network. If the cell is making lots of proteins, they'll be made by ribosomes through translation, but they'll be transferred to where they need to go via the endoplasmic reticulum, via the ER. Cells that produce a lot of proteins and then export them, send them out of the cell, send those proteins to a little structure called a Golgi body, named after the guy who discovered it and won the Nobel Prize for it in 1905, Camillo Golgi, Italian biologist. And you can think of the Golgi as being the cell's packing and shipping center. Proteins that are going to be sent out of the cell end up in the Golgi, where they get sorted. They may be modified in various ways, and they get packed up for transport. And then cells may also contain small little bubbles of membrane called vesicles, and or large bubbles of membrane called vacuoles that can be used for storing products or transporting them or various other things. There's a slice through part of the endoplasmic reticulum. What you're looking at is a whole stack of folded membranes, and you're seeing a section through them. So imagine I took a whole bunch of bed sheets, folded them neatly, and then sliced right through the pile. I'd get something like this. Each one of these lines is the edge of a membrane. And proteins can pass in between these folds of membrane to get where they need to go. And you might notice that on some sides right here, the one of the membrane surfaces looks kind of rough. Like right here, you can see that between those two folds of membrane, it's actually kind of rough looking. That's because this part of the ER is studded with ribosomes. And each one of those tiny little black dots is a single ribosome, like those. That's how small they are. And you find a lot of this in cells that make a lot of protein for export. You've got cells in your salivary glands right now that are transcribing a gene and translating a protein called alpha amylase. It's a digestive enzyme that breaks down starch. So cells in your salivary glands right now are making that protein, and that protein passes to, or that protein is made on ribosomes that are stuck on the rough ER, and it passes between these folds of the rough ER, ends up packaged in a structure called the Golgi, and then expelled out of the cell and goes into your saliva. So that's what I mean when I talk about a cell packaging proteins for export. Your salivary gland cells make a lot of protein and spit it out of the cell, and it goes through little ducts in your salivary glands and ultimately out into your mouth, enabling you to digest starches as soon as you, even before you've swallowed them. Some cells have a lot of smooth ER, and here's a slice through that. You'll notice that it's not studded with these little black dots. This part of it happens to look more like a network of tubes. I know it looks maybe a little confusing, but you can see various folds and tubes in the membrane. And a lot of enzyme activity might go on in the smooth ER. Your liver has lots of smooth ER because the smooth ER is where liver enzymes sit and work at doing things like detoxifying poisons. If you drink alcohol, the enzymes that break it down are ultimately located in the smooth ER of liver cells. Cells that produce a lot of fats or a lot of carbohydrates may also have lots of smooth ER. And then going back to the rough ER, I talked about your salivary gland cells that are constantly producing amylase enzyme. That's a protein. That protein passes through the folds of the endoplasmic reticulum and ends up getting passed to a structure that looks like a stack of flat sacks. The best model I can come up with is imagine you've got 10 deflated beach balls stacked one on top of the other. Or maybe like a stack of pancakes, except the pancakes are hollow on the inside. That's more or less what the Golgi looks like, or Golgi body. And what you're looking at there is just a cross section through the stack. Right, and each one of these, like this thing right there, that's one of these flat sacks of membrane, one of these flattened beach balls. Okay, anything that's secreted from the cell passes from the Golgi into these little bubbles called vesicles, which bud off of one side of the Golgi, move to the outer membrane of the cell, and fuse with it, releasing the contents. And we call this exocytosis, by the way. And again, it may be useful here to think of soap bubbles, because sheets of rubber or plastic, the bubbles in your bubble wrap can't do this, but bubbles that are made of a thin film of liquid can bud off and then fuse together. So what happens when a cell releases a protein is that one of these little vesicles, one of these little bubbles, comes right up to the protein, or sorry, right up to the outer membrane of the cell, and then bursts, and the bubble fuses with the outer membrane of the cell, and whatever was in the bubble gets spilled out of the cell into the outside. And again, this is constantly going on in all of your cells that have to export lots of protein. All right. So vesicles are really small. Vacuoles are bigger and usually can be seen with a light microscope. Some vacuoles will store products over the long term. Many protists have vacuoles that pump excess water out and maintain homeostasis. Cells may excrete through vacuoles, and these are plant cells. And this plant cell in particular has a huge vacuole right there in the middle. What that vacuole is doing is storing water. And as long as there's plenty of water in the vacuole, the cell is pretty rigid, because, if you've ever handled a water balloon that was inflated close to the bursting point, uh, you know, a water balloon like that can be pretty rigid, can be pretty firm. And a number of plants that mostly use water pressure in their vacuoles in order to hold their cells up and be stiff and firm. Great example of this: go out to the grocery store and have a look at the lettuce. Your lettuce, your spinach, all those greens are nice and crispy and firm because vacuoles in the cells still contain lots of water, and it's pressure from that water. The technical name is turgor pressure, but don't worry about that. Pressure from that water is actually keeping that cell pretty firm. If your lettuce dries out, then water is lost from that vacuole and your lettuce wilts. Which is why if you go to a typical grocery store, they have these automatic watering systems that spray water on the lettuce every hour or something like that. They want to keep it fresh and crisp for as long as possible, so they can't let the lettuce dry out, or the central vacuoles will lose all their water and your lettuce goes flaccid. Gods know we don't want flaccid lettuce. Okay, this shows there's actually more rough ER right here. So like right there and right there are membranes in the rough ER, and then there and there and there you have a whole bunch of ribosomes that are stuck onto the rough ER. But right in the middle of it, you've got this oval thing that has folds of membrane inside it, and this is a mitochondrion. And it was going around in a meme for a while about those guys from the reality show set in a motorcycle shop? Was it Chopped or Choppers? Yelling at each other and having an argument, and one of them is yelling, "Mitochondria are the power plant of the cell." And this has kind of become a meme for something useless that you learn in college. It is sort of true. You can think of mitochondria as a cell power plant, because what it ultimately does is convert a great deal of the food that the cell takes in into a form of chemical energy that the cell can use to power everything else that it does. I want to give you a better understanding than just mitochondria are the powerhouse of the cell. So let's move on. We just haven't got time for the details, but this is a molecule called ATP. And you might notice that it's a nucleotide. That happens to be the base adenine. That's a sugar. That is ribose, in fact. But instead of a typical nucleotide, instead of one phosphate group, it's actually got three. So that's why it's ATP, adenosine triphosphate. Three phosphates, triphosphate, like trisigma. The reason this might actually sort of matter is that there's a lot of energy that's stored in the bonds that connect one phosphate to another. So what ATP can do is give up one of those phosphate groups, break it off and stick it onto another molecule. So ATP becomes ADP, adenosine diphosphate, when it loses one of those phosphate groups. So ATP goes around sticking phosphates onto other molecules. And the reason that matters is getting a phosphate group stuck onto a molecule gives that molecule a kick of energy. What ATP is doing is transferring energy to the molecule that gets a phosphate group stuck on it. A simple example is you've got lots of ATP in your muscles. That's a lot of muscles. There we go. And in particular, one of those proteins is called myosin. Another one of the proteins is called actin, and your muscles contain fibers of myosin and fibers of actin lying parallel to each other, right next to each other. When a molecule of ATP gives up a phosphate to myosin, it causes the myosin to change shape for just a moment. And that shape change allows the myosin to slide past the actin. So you have these filaments of myosin and these filaments of actin. They slide past each other, and that's how your muscles contract. Every little twitch of your muscles is millions of actin fibers and millions of myosin fibers ratcheting past each other, click, 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 until the muscle is fully contracted. And they're able to do that because myosin can change shape, and it changes its shape and does this little click thing where it slides past the actin whenever a molecule of ATP gives up a phosphate and some energy to the myosin. So ATP is the fuel that runs metabolism, or at least the endothermic reactions of metabolism, the ones that need energy to happen. And most of the work of generating ATP is done by your mitochondria. They carry out most of the chemistry, not all, but most of the chemistry that's able to extract energy from food, and they use that energy to convert ADP back into ATP. In fact, every ATP molecule in your body gives up a phosphate to become ADP, and then gets a new phosphate stuck on to become ATP again, somewhere between 500 and 750 times every day. You're constantly breaking down ATP to drive your metabolism and then recreating ATP through some chemical magic that your mitochondria mostly do. And so you're constantly cycling your ATP If all of the ATP that you use up every day could exist at one time, it would be equal to your own body's weight. You'd burn your whole body's weight in ATP every day. That doesn't happen. What happens is you've got a relatively small amount of ATP, but you convert it to ADP and then regenerate it mostly in the mitochondria. Every molecule as many as 750 times each day. So now you know a little more about why mitochondria are the powerhouse of the cell. They do most of the work of generating the ATP that your metabolism needs to do an awful lot of stuff. Way back in lab 2, hopefully you saw chloroplasts. They're those green dots in the water plant leaf. Those are the organelles where photosynthesis takes place, harnessing solar energy to take water and carbon dioxide and build those into sugars and then into larger molecules that the plant needs. So that's the plant's little solar panels right there, are the chloroplasts, these little green dots. If we go back, yeah, this being a plant cell, each one of these things right there, that and also that, are chloroplasts. They've got lots of complex membrane structures inside them. Don't have time to go into more detail about that, but they're pretty complex things when you look at them. Locking everything together is what we call the cytoskeleton. Remember, cyto means a cell and skeleton means a skeleton. These are pictures of cells that have been stained so that the nucleus looks yellow. So there's the nucleus, there's the nucleus, there's the nucleus. And you can see that the nucleus of each of these cells is surrounded by what looks like flame red cotton candy. Kind of looks like, oh, I don't know, maybe, Judas Priest flavored cotton candy. That's a network of proteins that make up what we call microtubules and microfilaments. There's actually a third type called intermediate filaments. I'm not going to worry about those. Those support the cell, those give the cell shape. And if it's a cell that can move, like a white blood cell or an amoeba or some other protists, the cytoskeleton is what allows that cell to move. They may also hold structures in place within the cell, like an internal scaffolding. And some eukaryotic cells have hair-like structures that beat back and forth, little hairs that wag back and forth. In some cases, these enable the cell to swim and move through fluid. In other cases, like these cells from the lining of your windpipe, your trachea, the cells don't move, but the beating back and forth of these millions of little hairs pushes fluid past the cell. So they can either move the cell through fluid or move fluid past the cell. For reasons, if they're very long, we call them flagella. Flagellum is Latin for a whip. If they're short, we call them cilia, which is Latin for your eyebrows, right? Short hairs versus long hairs. But they've got the same basic structure. The only real difference is length. And that's a bunch of cilia right there from the cells that line your windpipe, your trachea. Last thing I'll try to cover. This has been kind of a lot, but a question you might be wondering is where in the world it all came from. Well, the answer that we now accept as being far and away the best explanation for how you could evolve something as complicated as a eukaryotic cell was worked out by Dr. Lynn Margulis, who I actually got to meet a couple of, maybe just that once, but we did have dinner. She's probably the single most famous scientist I've ever gotten to have dinner with. She published a book in 1970 called Origin of Eukaryotic Cells. Like every biologist that's ever been, she built on some earlier ideas, but she was able to back those up with data from this new field that was just then beginning to be called molecular biology, which is the study of DNA and proteins and transcription, translation, metabolism, all of that. She was able to back up these ideas with data from newly invented types of microscopes as well. And she proposed that eukaryotic mitochondria used to be independently living bacteria. They used to be organisms on their own. They penetrated an early eukaryotic cell 2 billion years ago next Thursday. Just kidding. Took up residence inside. Maybe they were originally parasites that fed off the eukaryotic cell, or maybe the eukaryotic cell tried to eat them and couldn't quite. We don't know exactly how it happened, but they started living together and became so dependent on each other that neither one can live without the other now. Your cell, you would die pretty quickly if you lost all of your mitochondria, and your mitochondria would die pretty quickly if we took them out of your cells. They can't live without each other. This type of relationship, by the way, is called a mutualistic symbiosis. Symbiosis means living together. Mutualism is a type of symbiosis where both partners benefit. The same, Margulis said, was true for chloroplasts. They're the descendants of free-living photosynthetic bacteria that entered early host eukaryotes and took up residence there and became mutually dependent on their hosts. Plants can't live very long without chloroplasts. Chloroplasts can't survive outside of plant cells. We have another mutualistic symbiosis. You'll also see this called endosymbiosis. Endo just means internal, inside. Crazy idea to think that, there is a sense in which you are not an individual. You are a whole bunch of cells walking around that all came from, your mama's egg and your dad's sperm back about 21 years ago, whenever it was. But key parts of your cells are descended from ancient bacteria that used to live entirely independently. You're actually a fusion of very different types of organism. Raises the question as to whether you're taking your mitochondria for a walk or whether it's the other way around. Sounds weird, but mitochondria and chloroplasts, we now know, have their own DNA. It's usually circular, like bacterial DNA, not strips like host eukaryote DNA. And the DNA sequences look more like bacterial DNA than they look like host DNA. Mitochondria and chloroplasts also have their own ribosomes. Bacterial ribosomes are a little smaller than eukaryote ribosomes, but mitochondria and chloroplasts generally have bacteria-sized ribosomes, not host eukaryote-sized ribosomes. Mitochondria and chloroplasts still have to make some of their own proteins. They carry out transcription and translation inside themselves, independently of what's going on in the rest of the eukaryote. And mitochondria and chloroplasts can be killed by antibiotics that kill certain bacteria. And if a eukaryotic cell loses them, it can't replace them. If all of the mitochondria in your cells died, you couldn't make any more. This right here is a map of mitochondrial DNA. Each one of these colorful sections is a different gene. We don't need to worry about the details here, but I wanted you to see that mitochondrial DNA is a loop. That's a bacterial feature. It's not something you'd expect to find in a eukaryote, loopy DNA. And if we do a cladogram based on DNA sequences, mitochondrial DNA is right here. These are 3 species of eukaryotes that contain mitochondria, and we use PCR, remember that from last week, to read the sequences of mitochondrial DNA from eukaryotes and compare it with DNA from bacteria, and boom, they share a common ancestor right there. Mitochondrial DNA turns out to be most like the DNA of a type of bacteria called rickettsias. Rickettsias can cause certain diseases like typhus or something called Rocky Mountain spotted fever, which you can get from tick bites. Not Lyme disease, that's something else. Mitochondria and chloroplast are surrounded by double membranes. You can see 2 layers there. And that makes sense if you assume that at one point mitochondria and chloroplast ancestors were engulfed by a host. So that's your big take home lesson right now, that the complexity of eukaryotic cells is in large part the result of 2 or sometimes 3 very different types of living thing coming together somehow and establishing a mutualistic symbiosis, a relationship that benefits both. And in fact, it's so beneficial that neither can live without the other now. Which makes me think about, I'm thinking of a eukaryote nucleus and its mitochondria singing that song by Queen. I can't live without you. I can't live without you. Hey, still got the high notes. Right, I'm going to stop recording right there.